G'day, we're going to be discussing one of the most important concepts or ideas in this soil carbon space. And this is the idea proposed by Dr. Christine Jones, which is the liquid carbon pathway. If this is the first episode you're watching in the soil organic carbon course I put together, go and check out the first few episodes talking about what is soil organic carbon, the benefits, as well as the different pathways, including the decomposition pathway. We'll be talking about the liquid carbon pathway and how it differs to the decomposition pathway and some of the benefits and disadvantages to the system. And as a reminder, go check out the course on our website, agresol.com.au. You'll find a whole bunch of additional resources and supplementary material for your enjoyment. There's no email opt-in or anything. It's completely free. Go check it out. Cool. So the liquid carbon pathway was first introduced by Dr. Christine Jones in a number of different publications. You can go find all those on her website at amazingcarbon.com. And really, there's four steps to this process. The first one is photosynthesis. We get our carbon dioxide from the atmosphere. Plants capture this, fix it into glucose in this amazing process called photosynthesis. It combines carbon dioxide with water, uses chlorophyll, sunlight to produce glucose, and a byproduct of that is oxygen, which we all get to breathe. So they take the carbon in the atmosphere, fix it. The second step is resynthesis. They take the glucose and they remake products from that glucose. So some of them can include starch and cellulose, which is basically just glucose stuck together. Proteins, fats, oils, and organic acids, a whole range of things and more. They make all these things, and then it leads us on to our third step, which is exudation. So the plant releases these sugars and soluble carbon molecules into the soil to feed and stimulate the soil biota or the soil microbes. So really bacteria and fungi, they're the main ones. Nematodes and protozoa usually eat the other ones. But it really is there to feed the rhizosphere, which is the combination of all those in a very small area around the root zone. You might see on like kind of dreadlocks around the root. That's what it is. Now, it's estimated that around 30 to 40% of the carbon fixed in photosynthesis is released through the roots to feed the rhizosphere. This is a massive amount of the effectively food source of the plant. Like they're giving away more, almost up to half of the amount of carbon. Now this is pretty typical. It can go higher, and we'll talk about how that can go higher later on in the video, where we talk about maximizing this process to really increase the amount of soil organic carbon we can fix. So stay tuned to that. So the plant releases these carbon molecules into the root and including into our mycorrhizal fungi, which form a symbiotic relationship with our plant roots. So if this is our plant root here, the mycorrhizal fungi actually goes into the plant root and colonizes it. So it's a really amazing beneficial relationship for both of the plant and the mycorrhizal fungi. And so some of the carbon goes towards our mycorrhizal fungi for a number of different benefits. So one can be the trading of minerals and water for this carbon source. But another benefit is that the mycorrhizal fungi fixes this carbon in aggregates with a whole number of different compounds and minerals through this process called humification. This is where we get a organo-mineral complex called humus, which is in our soil aggregates and builds these really stable soil aggregates in the soil. And this is how we build stable soil organic carbon as humus, which is the largest fraction of our soil organic matter, and super important system. Now from this system, about 46% of the root exudates, or the carbon in this system, actually gets pushed into soil organic matter. So that's a very high, conversion rate for this system compared to our other one, which was the decomposition pathway, which is about 8%. This is almost 6 times more efficient at converting carbon into soil organic carbon. Now that came from a study where the range of data it was 18 to 91%. So there's a massive range in that. And we'll talk about why that range is like that and what we can do to try and increase it up to that 91%. But effectively, this is a super efficient system that we can use to make soil organic carbon. Now, there's a number of benefits we get from this system. The first one is that it's super efficient at making soil organic carbon. It's rapid. We can get this process firing quite quickly and fixing carbon very quickly. So that's the first one, efficient and quick. Next one is that we can fix carbon to depth. So depth within our soil profile. How far exactly? Well, it really depends on how deep the roots go. We can build soil organic carbon as far as our roots go in the soil compared to the decomposition pathway, which is really just in the top 10 centimeters of soil, which is where the litter or our residue really stays. And I mean, the roots that decompose within the soil is pretty marginal, to be honest. But this system allows for root exudates to be produced, to be released throughout the soil profile as far as there's roots. So in a system where we have perennial plants with roots going deep into the soil, we can fix soil organic carbon really, really far. And this is really evident in this photo from Dr. Christine Jones's work, where you have soil organic carbon, or that dark soil, all the way down that profile, I think even past 1.2 meters. It's quite deep compared to the other side, which is just that top layer of soil. So we build soil to depth. It's efficient and easy. The other thing is that it's quite cheap to do. We'll talk about how we can do it, but it doesn't require us dumping all this compost or dumping all this biochar in. It just requires us to grow plants, which is what we should be really good at as farmers. Next, this humus is really resistant to decomposition from microbes as well as oxidation. So it's in a stable form. This stable form of soil organic carbon is a lot more active, biologically active as well as chemically active. It gives them a lot of structural benefits. And it also assists in building fungal networks and this exchange between our mycorrhizal fungi and our plant. And so there's mineral trades and water trades happening at the same time. So not only are we building soil organic carbon, but we're also improving our plant health as it has this symbiotic relationship with our mycorrhizal fungi. Now, there are some negatives that we need to talk about. The first one is that we need a mycorrhizal fungi network for this to function, which means we might have to kickstart the system, which might take a little bit of effort, and it might take a little bit of work to get this working efficiently. And a lot of our agricultural practices are antagonistic to this process. So we have to rethink how we manage our farms and how we grow our plants so that we can allow for this system. So now we're going to discuss ways that we can actually maximize the efficiency of this process to really drive the liquid carbon pathway. So I've divided this up into two stages. So we have the plant stage, which is all dependent on the plant, and then we also have our mycorrhizal fungi stage, which is all dependent on mycorrhizal fungi. So we'll start with the plant phase. So ultimately, we are looking at the amount of photosynthase that the plant can fix and then pump into the ground. And ultimately, that's really what the plant is required to do on this. And so there's two ways we can do this. The first is by increasing root exudates of lipids. It's estimated that humus is made up of about 40% lipid or like fatty content, which means if we increase the lipid component of the root exudates, then it increases the energy that the mycorrhizal fungi have to then convert that into humus. Now, in order to increase lipid content, plants are required to increase their photosynthetic rate first and then their protein synthesis rates before being able to increase their lipid production. So a lot of this is nutritional in nature, and in order to increase our photosynthetic rate, we need to make sure that we have sufficient amounts of minerals and nutrients that are required for photosynthesis. So I got a whole video on increasing photosynthesis in plants. You can go check it out. And we talk about a lot of these, but the minerals are nitrogen, phosphorus, sulfur, magnesium, iron, and manganese. All of those are used in photosynthesis. Now, that can either be applied as a seed coat just to start things off, Otherwise, you can apply that as foliars. And it is better to apply these as foliars, especially our trace minerals like manganese and iron, as typically when it hits the soil environment, they get locked up very easily due to redox reactions. And so when we apply these as foliars, it rapidly moves into the plant. To know whether or not our plants actually have these in sufficient levels, we can do a sap test or a tissue test, which can have limitations. Soil tests aren't the best for understanding exactly what's in the plant. A sap test, or even better, differential sap test analysis would give you the best idea. So what this will do, this will really increase the rate of which photosynthesis will occur, or the rate of which carbon is fixed into the leaves. Some estimate that plants are really only photosynthesizing at around 20% of their capacity. Once you get this nutrition right, it can almost go up 5 times. Now, a lot of the times, you're not going to get 5 times as much growth. Growth will cap out. What will happen is that all that energy or all that carbon will start being pushed through root exudates. So it's not just 40% of the root exudates, of the carbons being released through root exudates. It's going to be more like 80% of the overall carbon fixed by the plant will be contributing to root exudates. So as our photosynthesis go up, a greater proportion of root exudates are given out to the microbes. As I was saying, plants prioritize protein synthesis before they get to lipid production. In order to maximize protein synthesis, we need sufficient amounts of nitrogen, magnesium, sulfur, boron, and molybdenum. We've got a whole video on all of the minerals, also on the YouTube channel, so go check them out. We also want to reduce our applications of pesticides, including herbicides. This inhibits the protein synthesis pathway. So for a cover crop, for example, where you don't really care if there's too much pests, because we're not trying to pull off a crop, just don't apply any pesticides or herbicides. Cover crops, and specifically cover crops, you just let it do its thing and you can terminate the whole thing. There's really no need to apply herbicides until the end, in which case you're terminating it. So finally, we want to increase lipid production. So lipid production requires a lot of energy, and the plant can't get all that energy if it's obtaining its nitrogen as inorganic forms, because it requires a lot of energy to convert nitrogen from an inorganic form into an organic form. Instead, all of the nitrogen needs to be obtained as either amino acids or amino sugars, and that can be done with symbiotic relationships with microbes, typically at the rhizophagy cycle. The more nitrogen the plant can get from organic sources, the more energy the plant will have for lipid production. Once it reaches that stage, it will increase the overall root exudates as well as the component of lipids in that root exudate for the mycorrhizal fungi to use. Now finally, in this, with additional plant diversity, we can increase the overall photosynthetic ability of the area with varying canopy architecture and root architecture, making sure the whole kind of ecosystem is running efficiently for that either cover crop, pasture, and even cash crop. Next, we want to make sure that we have something growing all year round. Because if we only have a plant growing for effectively 4 or 5 months, it doesn't matter how well the plant's photosynthesizing, the rest of the year it's all barren. So implementing regenerative grazing techniques, so AMP or holistic management grazing techniques, to make sure you have as much green biomass as possible in your pastures. We want to, in our pastures specifically, we want to have a combination of C3 and C4 plants so that we have green plants growing across summer as well as winter. And then in more of a cropping situation, using cover crops instead of fallow, or at least even part of our fallow is cover crops. Utilizing pasture cropping potentially. We could also look at relay cropping, a whole different type of, a whole range of different techniques that we could use to extend the photosynthesis of an area more than just our typical 4 or 5 months of the year. Now the benefit of that is we will produce root exudates all year round to feed mycorrhizal fungi to produce humus. Now on to the mycorrhizal fungi stage. Effectively what we want is a strong, robust mycorrhizal fungi population and community with hyphae going all through the soil. And so typically everything that's good for soil health will be good for mycorrhizal fungi health. Things like limiting our chemical use, reducing obviously fungicides. Obviously if we want to promote a fungi in our soils. We want to minimise fungal use, as well as salt-based fertilisers and a few other minerals like nitrogen and phosphorus can inhibit mycorrhizal fungi development and colonisation of the root. We want to make sure we're practicing no-till. If you think of fungi as a network of hyphae throughout the soil, if we're ripping them up, that's not going to be good for the fungi. We'll make sure we have ground cover. That's good for just microbes in general. Very important that we have a living root. So mycorrhizal fungi do not live on by breaking down organic matter. They live exclusively in a living root. So they have to have a symbiotic relationship with a root in order for them to survive. So if you don't have that living root, they start to die off. My understanding is that they can start to die off even within like 2 weeks of not having a plant root there. So making sure we have systems where we're using cover crops, pasture cropping, or regenerative grazing techniques that extend the growing points of our plants to make sure that we have a living root in the soil all year round is very important for maintaining a mycorrhizal fungi. Finally, if we have a history of a lot of fallow or long fallow, adding a mycorrhizal fungi inoculant is quite important just to get those populations re-established, and then biostimulants to stimulate the overall soil biota. So all of these things will help our plants to fix more carbon in a really rich energy form as a lipid, and then give them to our mycorrhizal fungi to then produce humus all year round. And again, this is the number one way to build soil organic carbon. It's not by adding compost or biochar, even though they can be great for different things. If you want to build soil organic carbon, this is the best way to do it because of the high carbon conversion efficiency of 46%. Super high, so much better than the other system. So if you like this, make sure to subscribe. We've got a whole range of different videos that is just out there for people to learn more about agriculture. Make sure to check out our website as well. There's a whole range of additional resources that you can get for free. No email opt-in at all. It's all for free. Otherwise, my name's Teal. Thank you for watching. Cheers.